Yeah. Oh, it kind of looks. It's not, yeah. And also. That's the quote from the letter.
Okay.
They said that and take it out of context.
Okay, let's get started. Maybe a minute earlier. All right, you have question? Yeah, my bad. And I messed up. Who emailed me? You. It's the 17th. In my email said the 18th. I don't know what I was thinking. Yeah. That is based. I didn't come up with that. It's not. Yeah, the university randomly selected dates and time. I'm sorry. I don't know if I can move it half an hour.
Yes.
If you can, please do.
I can. I'm gonna be here at 8. Come whenever you want. If you want to come, but not later than 8:30. So, yeah, we have the room. We should have the room. I don't think there will be anything right after us. Yeah. All right, but I'll be here at 8 for those of you that want to come early and be done.
Yeah. Up in 6.
Yeah, whenever you come until 8:30, you'll get the 2 hours. Okay, so if you come at 8:30, that's fine. Okay, so Tuesday the 17th. Thank you, Kendall and Alicia, for noticing that. Okay, don't forget the review session on Wednesday in lab at 9:30. So please be there. It's very important. We're going to go over a lot of questions and topics. And a lot of these questions will show up in one form or the other in the format. So it's essential that you come. And I have those questions on Canvas. So start looking at those to be prepared. Okay. Any other questions? All right. We can go over on Wednesday also ideas of how to study for the finals and what to focus on. So I'll cover that during the review session. Okay. Good. All right, just quick recap question. What is protein separation utilized for? You remember anything after a long weekend? Was it a long weekend? And no lab? Yes, so we isolate protein to identify its characteristics and learn more about it. What were you going to do? Yes, so part of protein discovery is isolation and identifying its properties, but a very big, important industry related. Yes, so we do have to isolate the general functionality, but in terms of industry, what is separation important for? Concentrate production of ingredients, production of protein ingredients through separating the protein from complex matrices. Okay. What are some methods of separation we talked about? Do you remember? We talked about, yes, somebody? Adsorption, yes, method of separation using chromatography based on the characteristic of the protein and its adsorptive properties. Very good, chromatography, adsorption. So sorting out and then you dialyze, you correctly remove the salt. Ion exchange, that is chromatography based on adsorptivity, correct. What else is we can do? We said solvent extraction. So we can use organic solvent to precipitate the protein. We go even by size. By size, very good. Size exclusion is one chromatography. Membrane separation by size is another. Were you going to say something else? Did I see a hand up? Well, basically separation by size. Oh, there's one that you didn't mention, pH, isoelectric point separation. So by change differences in solubility, by differences in size, by differences in absorption properties. So remember when you see a question in the final, and if I give you 3 characteristics of a protein, and I ask you how to separate it, you want to list 3 separation methods, 3-dimensional separation methods. If I give you 2, you list 2. So if I give you a charge and a size, you have to list something based on separation by size and separation by charge. Okay? So the hint is in the question. Okay. All right, here's where we stopped last time. I want to talk to you about electrophoresis, which is used to separate protein, but this is not to collect fraction. Usually we separate protein to characterize qualitatively the different components within the protein. So there are 3 types of electrophoresis that are common. One is called SDS PAGE, polyacrylamide gel electrophoresis. That's what PAGE stands for, polyacrylamide, because acrylamide is the material that the gel is made up of. So a matrix of acrylamide cross-linked, and then a gel and electrophoresis. SDS, it's called SDS because you put a chemical called sodium digoxide sulfate. You don't need to remember that. SDS for short. What it does is it interacts with the surface of the protein and it makes the protein carry a negative charge. So all the protein in SDS will be negatively charged. When we put them on a gel, so usually we get a gel in a cassette already made and ready. And it doesn't smell so great. But so there is a cassette. It's like 2 layers. You have 2 layers here of plastic. Inside of it is a thin gel made of acrylamide, polyacrylamide. And it has a kind of a cone. And for, has anybody done electricity in labs, right? Or not in class? Biolab? Biochemistry, yeah. Do you have a biochemistry lab? No, in general biology. Okay. General biology. So when you want to load your sample, you remove this cone and you use a very, very thin pipette to pipette in your protein sample. And you run, what you do is, first of all, you have to remove this tape so that the current can flow. You put it in a special compartment, and then you put running buffer in there. You put running buffer, you wear gloves because your hands have protein in it, it will contaminate the gel. I'm not wearing gloves anymore because I'm not running this gel. Once you put your solvent, you can gently remove the cone and load your samples in every well. And you run a voltage, so you put this here so that you attach it to something that gives you current. What do you call that? Power voltage thing, whatever. And then you let, what happens is, what did I say? Did I say anything bad? Okay. So you let it run, so the protein will all move in one direction. So you have the negative to positive. So all the proteins are negative. So the current goes and then it goes, all the proteins go from negative to positive down the gel and separate based on size. Because now you have the gel that have the 4 sizes, and then the bigger proteins will be at the top and the smaller proteins will go to the bottom. And then you stop when the protein front leads towards the bottom, you turn it You turn it off. Take your gel out. I'm going to take even this out now. And my students don't know this. I found out that you have this compartment. Did you know this? I showed you? Okay. So see this? This is especially made for you to put this and push down to break it open. At least Anastasia didn't know because she gave me a tool to open the gel, to open this. Like, what is this tool? You didn't know. I used to train my student and myself in labs, and now you guys train each other, so some things are lost. It's so cool. I love it. These are actually my hands when I was a postdoc, just that you know. Okay, so can you see this? The very thin gel. There's a very thin gel. So once you run it, you don't see the protein yet. You need to stain it. So you take it out. Well, it depends on, this is a bad gel, probably it will tear, it's an old one. But you try to take it out very gently so that it doesn't, sometimes you need to put a little bit of water. Yeah, they gave me water, like, so that it helps it come out. So it's very cool. And then you put, and then you put it in a container, you add a staining solution that will interact with the protein only. And then you destain, and then you look at your protein components. Very nice. You know, acrylamide is bad for you. Did you know this? What does it do? Causes cancer. You want to get it? Not when it's gelatin in gel form. So now it's safe. When it's powder or liquid form, you don't want to be doing it. So either that or I don't care, I'm old. I don't care if I die of cancer. So, but no, it should be safe. I have another bad joke. I already have cancer. You didn't know, did you? Okay, that's fine. Forget it. Am I recorded? I shouldn't have said that. Anyway, so it is fun, you guys. Another form of polyacrylamide gel electrophoresis is using urea. Instead of SDS, if you use urea, you can separate your proteins based on charge to size ratio. So instead of just separating based on size, you can separate based on another characteristic. Native page, nobody really uses it. You just put the protein in a buffer and run it. It's very hard to see anything because the proteins will be interacting via hydrophobic interaction, non-covalent and covalent, they won't separate. So you won't see much. So it's always often is best to use some reagent to help better in separation. Okay, so 2D electrophoresis is done too. So what we do with 2D is we run two-dimensional, what it means. The first dimension is separation based on charge. So you also get a very specialized gel cassette, but small, like a strip. And then what you do is you load something called electrolytes or amphalites. So they have different charges and you run a current. So they migrate those charges based on their load of positive or negative. They migrate and settle because you have a current running from negative to positive or positive to negative. So you generate what we call a gradient of different charges. Then after you run those amphalites, you load your protein and run a current. The protein also has charges. So what they do is they migrate and stop at their isoelectric point. When they reach a place, a gradient where they carry no charge because of the alpha-lights, they stop moving. So now we have the protein separated based on charge. Then what you do, you can take that little strip and then you get a very special cassette where you don't have wells, you only have a nothing, just one big opening, and you put that strip in that opening here, and you will have in there another gel that will separate based on size. And you run a current. So now the separated proteins based on charge will go down your gel and separate based on size. So look at this blue, for example. This blue is proteins that had similar isoelectric point, but when we ran them on size gel, they are separated. So you are enhancing the separation of proteins. I don't know what you guys are laughing about. What else did I say? Inappropriate? Did I say anything inappropriate? Okay. I'm being recorded. All right. So this 2D electrophoresis, usually we do it to enhance separation, and especially if we're doing later on the next lecture will be in immunoassays. If we're doing Western blots, are you familiar with Western blot? Some of you might have heard of it. I'll talk about it in next topic. You can look at those antigens specific that interact with antibodies. So the better separation you get, the better sensitivity of that Western blot assay. So 2D is often coupled with Western blot. Okay, I'm gonna skip this. You can put on your slide skip, don't study. That's because of time. Capillary electrophoresis, it's okay. Protein nutritional quality is characterizing the how good this protein is for us from an amino acid perspective and digestibility perspective. So it's very important to make sure we have the essential amino acids that we need, and also that the protein we can digest and get the benefit of. So that's what the nutritional quality is made up of. Two important things: amino acid composition, digestibility. And there is something we call first limiting amino acid. Do you know what first limiting amino acid means? Have you heard about it somewhere else? Kendall. So first limiting, that means if you have 8 essential amino acids or 9 essential amino acids in a protein, the one that is present in the lowest amount. So the essential amino acid in the protein that is present in the lowest amount compared to the other essential amino, that becomes the first limiting amino acid. And nutritional quality is also impacted by anti-nutritional factors that impact digestibility. For example, enzyme inhibitors. If you have trypsin inhibitor in soy, for example, it will inhibit our digested protein trypsin, and the protein won't be digested as well. That's why soy product has to be heated to inactivate trypsin inhibitors. Lectins, for example, are not good for us. There are proteins in beans. If we consume raw beans, it might cause inflammation in our gut. Phytates, what do phytates do? They bind calcium and reduce the bioavailability of some of the minerals, including calcium. Saponin, tannins, they bind to the protein and they limit digestibility. So these are examples of anti-nutritional factors. So how do we determine amino acid composition? We hydrolyze the protein by acid or alkaline to break all the proteins into singular amino acids. Then we run them on chromatography, ion exchange, or reverse phase chromatography. And we run standards and we quantify each amino acid. Each amino acid does not absorb light by itself. Only 3 absorb light: the tryptophan, tyrosine, and phenylalanine. So in order to be able to detect everything using a UV detection, we do something called derivatization with a compound that will give us a product that we can measure light absorption. So So often we do reverse phase coupled with UV detection and derivatization to measure all the amino acids. And then from there, we get the amino acid score. Once we have the amino acid composition, we get the score, which is the milligram of the first limiting amino acid in 1 gram of your test sample compared to a reference sample. Usually casein is the reference. And we get a score. We use this score to determine something we call protein digestibility corrected amino acid score or protein efficiency ratio. These are the most common protein quality tests. So the protein digestibility corrected amino acid score, we need the amino acid score and we need protein digestibility. So how do we measure it? In vivo is the one that we need for nutrition label. There are many in vitro assays. We do actually in the lab, our lab in vitro assays so that we don't need animals models. But for nutrition labeling, to calculate % daily value, we are required to do the in vivo assay, which involves rats, unfortunately. So you have to get rats. You have to feed them a specific diet with a specific amount of protein, and you measure true digestibility. So how much protein they were fed, how much nitrogen they were fed, and then you take their feces and measure nitrogen in the feces. And then you will be able to determine how much nitrogen was actually absorbed. And you calculate the true digestibility, you multiply it by the score, you get a PDCAS value from 0 to 1. 1 is the maximum. You use that PDCAS to determine the daily value. How? You take the content of the protein in your sample, you multiply it by the PDCAS, and then you divide it by the requirements per day, the 50 grams requirement per day. So, for example, if your nutrition bar has 10 grams of pea protein, but the PDCAS of pea protein is 0.8. So what is the daily value of that? So you would say you have 10 grams in your serving, and you have a PDCAS of 0.8, and then 50 grams is your daily requirements of protein. So 8 over 50, what is it? What's 8 over 50? Calculate it quick. I don't have that. 0.16. Yeah. And then if you wanted a percentage, then you're getting 16% is your daily value. This is how you calculate your daily value. How much protein you have in your serving by the PDCAS divided by your requirements per day, and you get your daily value. So PDCAS, quality of the protein is very important to determine the daily value. So you don't take the total quantity of protein because not all of it will be digested. Not all of it might not have the complete amino acid, essential amino acid. The other one is the protein efficiency ratio. This is the method they use in Canada, but it's also used in the US only for infants under 1 year old. Why is that? Because it is based on measuring weight gain. So, of course, when we eat protein, we don't gain weight by just eating protein. So it's not really good for determining the nutrition quality for adult foods. But for infants, when they eat protein, it's associated with their protein intake. So in this assay, they get baby rats and they feed them protein and measure their weight over. And that's how they determine true digestibility. And the baby rat's growth rate is equivalent to human infants between 0 and 1. So, interesting facts. Other nutrition test is calculating an index for all essential amino acid, doing the in vitro protein digestibility assay by measuring digestion with over time and measuring pH drop, because when you hydrolyze protein, you release carboxyl groups, so the pH goes down. Another one that is important is looking at lysine availability. Lysine is an essential amino acid and it is destructive during processing the glomerular reaction. So by measuring lysine loss, it's part of an indication of the nutrition quality. So you have an assay that is multiple assays, but one common one is FDNB reagent that reacts with the lysine, and then you measure using spectrophotometric method. So in your book chapter, there are sections on assessing protein structural properties and protein functional properties. So these are essential to characterize new protein, to understand how proteins differ in a food matrix, different protein, whey protein versus soy protein versus pea protein. So you do structural analysis and then you do functional analysis. So these are FYI. I'm not going to ask you about them, but there are sections in your chapter if you want to learn more about them. Any questions before moving into a different topic? Well, it's not very different. It's in the realm of proteins. There is a question? Ah, sorry. Hi.
So why does Canada do it?
You know, it's Canada. I don't know. I don't know why. It's just been a question for them for years. Now there is a new method called DIAS. Have you heard about it? It's actually, it takes into account the potential digestion of proteins by microbes in the large intestine, which PDCAS doesn't take account for that. So, and people in Canada are pushing for that one to become the official method. So they go from not that great of a method to DIAS. It's a good move. All right, so now, what do I want to do? They changed, I had to update the computer, so they changed how we go to slideshow. It used to be an icon over there. I don't know what happened. There. Immunoassays. What are immunoassays? When you hear immunoassays, what does that mean to you? Immunoassays. You too. He's smiling the whole time. Okay.
And it looks like those tests for antibodies.
Yes. So immunoassays use antibodies, antigens in that. So the reaction of antibodies with antigens, and most of the times these antigens are proteins. There are other antigens that are not proteins, but it is the reaction, the assays are based on interactions of antigens with antibodies. So the antibodies are made up of proteins, actually. So if you look at that, okay. I didn't do that either. So what happened? Help. I don't know what happened. Okay. So antibodies are made up of 4 protein chains. So you have the blue chains that are identical, and the longer ones, bigger ones, and then you have the pink chains here. They're not in In reality, they're not pink and blue, but so you have them identical as well. So they are made of 4 polypeptides, these antibodies. And here at the top is the binding site. So this is the binding site that binds with the antigens. And this antigen here binds at an area we call epitope. So on the antigen, the area that binds to the antibody called epitope. So the antigen, what is an antigen? It's a foreign body that the body identifies as foreign. So people who develop allergies, how do they develop allergy? They have susceptibility to be allergenic to peanut, for example, or sesame or whatever. So how does it happen? They consume the peanut one time. Their body recognizes this as something foreign. Then specific cells in the body produces antibodies to that particular foreign body that is not usually foreign to me, but maybe foreign to somebody else. So what happens is the next time that individual consumes peanut, they will have a flare because they got sensitized from the first time they consumed it, because their body recognized it as a foreign body and produced antibodies to it. So in food analysis, we use that concept that there are antigens and antibodies, and we benefit from that for analyses. So the interaction between antibodies and antigens are very specific interaction and a very strong one, but non-covalent. Okay. So this is the actual shape of the antibody, potentially projected, of an immunoglobulin G or immunoglobulin. There are so many different types of immunoglobulins, but this is immunoglobulin G. So the affinity between antibodies and antigen is very, very strong, but non-covalent interaction. That means ionic, hydrogen bond, hydrophobic, van der Waals, and the affinity is really high. The affinity constant is really high for these, and very, very specific. Very high affinity and very specific. So the most important part of the antibody is this binding site, and for the antigen, the epitope, the site where it is specific for binding. So in proteins, for example, we have something called linear epitope and conformational epitope. What means that these are amino acids, 1, 2, 3, 4, 5, that means in sequence with each other. And these can be an epitope for a specific antibody. But the protein folds, sometimes the folding generate a specific epitope, 1, 2, 3 here, and it continues on the other side. So we call this a conformational epitope. So these are the illustrations for the linear and conformation. But this makes them very specific to binding for specific antibodies. So the size of this region is equivalent to 7 glucose units. The other important thing I want to point out is polyclonal and monoclonal antibodies. Polyclonal, I'm going to draw this so that you can visualize what I mean. This is important for assays, as I described when we were. Polyclonal, so I usually draw the antibodies as Y shape. This is an antibody, this is an antigen. This is semi-simplistic way of drawing. So if you take a protein, this is a protein, you might have one epitope here, one epitope there, one epitope here, here. What does that mean? That means that this protein has allergenic sites, multiple allergenic sites on it, that different antibodies can bind to. So different. So this is A, B, C, E. This is polyclonal. Usually, in a body, in the blood of a person that has allergies to peanut, if you take the blood and separate the serum, it would have polyclonal antibodies to peanut protein. That means in the serum, you will have different types of antibodies that bind to different regions of that peanut protein. So that's polyclonal. But in labs, you can actually produce one kind of antibody to one epitope. In lab, you get specific B cells and you activate those cells to generate one type, monoclonal antibodies. Why are these important? Because you can use them in assays. You can buy immunoassays kits, and these will have monoclonal antibodies. So these assays can be beneficial when you're doing screening so that you don't have to get humans or animals and get blood and get your antibodies that way. Okay, so that is important because buying kits, most of the kits or all of the kits out there contain monoclonal antibodies. So what are immunoassays? Why are they very helpful? They are simple, specific, and very sensitive. That means specific to specific antigens, very specific. Sensitive, they can detect small changes and have low threshold. And simple assay, not complicated, especially if you can buy a kit. But you need to know your antigen and you need to know what antibodies to buy. That is, or to work with. That is a very important criteria. And you need to be able to separate the antigens that bind to the antibody from other components. So that is a criteria, and I'll talk about it in a minute. And the immunoassays rely on a very important characteristics of the antibodies and the protein and antigens is the hydrophobic surfaces, and I'll explain that in a minute. So if you want to get polyclonal, that's the question here. If you want to get a polyclonal antibody to measure the allergenicity or the interactions for a specific protein, you want to cover all of the potential epitopes on the protein. So you want to get polyclonal antibodies so that you can determine whether that allergen is present in your food or not. So monoclonal might not work in that case. So what do? You take that antigen, you inject it into an animal, say a rabbit or a goat or what have you. And then you sensitize that animal, and then you take the blood. The serum will contain antibodies, polyclonal antibodies. That's how you. So, what are some applications for immunoassays? I just said one though, right? What was I talking about? Peanut butter, peanuts, allergenicity, to determine allergenic proteins. What else have you heard using immunoassays? Did you hear of anywhere else immunoassays are used other than allergy? Have you heard of it? Yes. Yeah, so it's used in microbial detection. So bacteria and viruses, very good. What else? GMO, genetically modified organisms. So if you want to determine if that whatever crop or ingredient or food has a genetic modified organism, you can check using immunoassays the presence of proteins from GMO. Advanced glycation end products, yes, because they're mostly polymerized proteins, yes. Very good. Another one Have you
I've got
heard
one.
of the case in like a few years ago where there was horse meat in the what? IKEA? In IKEA? I thought that was in England. I don't know that they did that in IKEA specifically. But there was pork patties, and it turned out to have horse meat instead of pig pork meat. So it was, you can detect not supposedly their meat species in your meat products by ELISA, and I'll show by immunoassays, not just ELISA. I'll show example later on. Toxins. So aflatoxin, for example, you can use ELISA to measure toxins and specifically ELISA. So protein detection, allergen, meat, species, authentication, GMO, toxins, identification of bacteria and viruses.
So when you talk about polyclonal and monoclonal, do the polyclonal, are they actually made like a bunch of different antibodies?
Yes, so that means they have different binding sites that could bind to different places on the protein. So polyclonal, that means a mixture. If you get a serum, like a serum from another individual, it will have multiple different amino antibodies in that mixture.
When you say epitope,
Epitope. So this
that's true.
is an epitope. One epitope, 2, 3, 4, a region where the antigen can bind.
is it just on the protein or is it also on the antibody?
The antibody has a we call it binding site. The binding site on the protein, we call, or on the antigen, we call it epitope. It's just nomenclature. You can also say binding site, but epitope is specific to the antigen. Okay, so the different immunoassays we're going to talk about, ELISA, which is the enzyme-linked immunosorbent assay, ELISA for short. There is sandwich ELISA, we're going to talk about indirect ELISA, competitive ELISA. We're going to talk about all these different forms. And then we're going to talk about purification and other ways of using immunoassays. So immunoaffinity purification is chromatography, lateral flow strip. Have you heard of those? Okay, I'll talk about them later. And Western blot, I'll explain it as well. All right, so enzyme-linked immunoassays, by the name, you have an enzyme. And the enzyme is there specifically to help generate a product with color. So then you can measure the presence of antigen. So the enzyme is linked either to the antibody or to the antigen. Two common ones are horseradish peroxidase and alkaline phosphatase. Why are they used? Because they are effective, quick, high activity. It gives us this colored compound from the substrate to be able to measure using plate reader. So oftentimes in ELISA, we have many samples we're testing and standards and a lot of that. So instead of like you did in the lab, one tube at a time, you have a plate with 96 wells. You can put samples as many as you can. You can get pipettes specific with multiple channels. You can pipette at the same time, multiple samples. And then you would run the assay. 96 wells at one time, they go into the reader, and then you would have, in a few seconds, absorption in all of these different wells. So the standards, the samples, everything. So it's very, very effective. Have you seen a microplate reader? Nobody used it before? It's very effective. We do have it in our lab. Oh, thank you for monitoring the chat. Ah, there you go. I wish. For what? I'm not sure what I was
He wishes
asking.
he's seen one.
Huh.
You actually seen one.
Oh, okay. All right. So what did he say? Okay. All right. So ELISA, we can run it in a direct detection or an indirect detection. So now this is where it's going to get a little confusing. So let me say it very slowly. With direct detection, so what that means is you have an antigen and you have an antibody that directly binds with the antigen. And this antibody is labeled. So that star is labeled with the enzyme. Okay, so the star is the enzyme. The label, we call it label here, but that means it's the enzyme. So this is direct detection. The more antigen you have in the sample, the more antibodies will bind, the more enzymes you have that will convert a substrate into a product, and the more color you get. So direct. So it is direct. You have more, the more antigen, the more color, basically. Because the labeled antibody is binding to the antigen that you're trying to detect, and the enzyme will react to the substrate, give you a color. Now, the indirect ELISA, you have, and here goes, listen carefully. You have antibody to the antibody. What does that mean? Antibody to the antibody. So if I have human antibodies here, so if I'm detecting allergenicity for peanut butter using human antibodies, okay? So this is the blue light, the blue is a human antibody, and this is the protein from peanuts. They're interacting, but this human antibody is not labeled. So what I have here, I can take the human antibody, inject it in a rabbit or a goat, and make that rabbit or goat sensitized to the human antibody. So now, antibodies are proteins, huh? So now I have an antibody that I get from rabbit, let's say, that is going to detect the human antibody. We want me to repeat? So again, with the example of antigen, we need peanut protein. And this antibody, I got it from a human serum. That human is allergic to allergic to peanut protein. So this is going to bind to my antibody. It's very hard if I'm getting antibodies from human serum. It's very hard to attach an enzyme to it. The experiment will be very long. It's easier to get an already labeled antibody with an enzyme that I created, the industry created in an animal. So how they create it in an animal model is they get the human antibody and inject it into an animal, a rabbit or a goat. So that rabbit or a goat is going to produce antibodies to that foreign material, which is the human antibody. We call it secondary antibody because it is not antibody to the antigen, it's antibody to the first or primary antibody. So that's why it's called secondary, and it is the one that is labeled. So it's indirect because it's not attaching to the antigen, it's attaching to the antibody. Why do they do that? Because it's easier to produce industrially a labeled antibody that we just buy when we're doing the assay, instead of us labeling the antibody that we get while we're doing an experiment. We get antibodies from humans. It's very hard for us to label it. It takes a lot of time. So we better buy antibodies labeled that is produced that will attach itself to the human antibody in this case. Okay? So that is what is known with indirect, because that it's not binding. The labeled antibody, secondary antibody, is not linked directly to the antigen. Any questions, clarification? So main steps, and I'm going to draw the main steps. This is important to know. And this is where the hydrophobicity of the antibodies or the protein antigen comes into play. So when we use a plate, microplate with 96 wells, that material, plastic material, is a hydrophobic surface. So here I'm going to give an example of a sandwich ELISA where I bring in antibodies first. So the antibodies are going to bind via hydrophobic interactions to the surface. So that's the first step, is bringing in my antibody or antigen and then have it bind via hydrophobic surface. Then you'll see there is intermediate washing steps. So I would have a wash that will any antibodies that did not bind will be washed off. The next step is putting a blocking agent. You know, so you have a lot of surface here. This surface, you don't want your antigen to unspecifically or inspecifically bind to these open areas. So you add bovine serum albumin, which is a protein, not of interest to your assay. Bovine serum albumin protein from bovine cow. It's a protein, so it's going to populate this region, the empty region. So you don't have non-specific binding. You prevent non-specific binding. So that's the second thing. And then we do a wash. Intermediate. So any other remaining proteins will be washed off. That is next. You put your antigen. And then the antigen will bind. And then you wash excess. And then your next antibody, that with sandwich. And you wash. Then you put the substrate for the enzyme, because this is going to be enzyme labeled. And then you wash. Then you put this, then that's it. Because the product will become, the substrate will become product. And then you measure absorbance. So these are the steps, and this is the illustration of these steps. So I will stop here, apparently. On Wednesday, I'll take some time of the review session to finish this lecture and then continue the review session. It shouldn't be a lot left. Okay. Thank you, guys. I'll see you on Wednesday, 9:30 in the lab. No, review from 9:30 will end at 12:30 because there is a lecture like usual in here in class.
I think great. Yeah.